This video will describe and show you how to perform meta-analysis in Jamovi with an outcome that is continuous and the estimated effect size is standardized mean difference. And when the outcome is continuous and the estimate effect size is based on correlation coefficient abbreviated as R, and when the outcome is binary and the estimated effect size is based on odds ratio or risk ratio, and also will describe how to perform proportional meta-analysis in Jamovi and how to generate a forest plot and a funnel plot. If you liked the video and found it useful, do subscribe to the channel and click on the notification bell to receive the updates and the new releases on statistical method for several statistical softwares such as SPSS, Excel, Stata, Jamovi, R Statistics, GraphPad Prism, the G-Power, GMP, and revaman. And click on the like icon and share the video with others to benefit and to support us to continue developing the channel and introducing and producing and publishing new videos and how to perform several different statistical method in any of these statistical packages or softwares. Meta-analysis in Jamovi and drawing a forest plot and funnel plot. Meta-analysis is a systematic review, literature review, and statistical method that combine and summarizes the results from several independent studies which investigate the same research question. So the analysis will provide precise estimate of the effect size of exposure or treatment or experimental compared to a control if the setting for that research was in that format. And the meta-analysis will pool all the results from multiple studies and then will generate an overall estimate effect size. The meta-analysis will analyze data from previous already research studies that has been done and pull them together to identify any common patterns and make a conclusion about the research study and whether there is a difference or an effect between the compared two groups, such as experimental or treatment or exposure to control. The meta-analysis will measure the variability of the study results. If there is any variability between the each research study from another, and calculate the confidence interval for each study and perform an I-square test. I-square test or homogeneity test, or sometimes it's called heterogeneity test, is a test that will measure the degree of variability or inconsistency in the result among the multiple studies in meta-analysis. The value for I-square is expressed in percentage An I-square value with 0% indicates no heterogeneity, and an I-square value of 100% means maximum heterogeneity. The meta-analysis will also generate a forest plot. Forest plot is a graph that represents the results from meta-analysis with summary attached. The plot consists of three major parts. The first part is a statistical analysis and statistical description for each study with the number of participants and the estimate size. The second part is the diagrammatic representation of the data output, data analysis output, statistical analysis, and the third section is the overall estimate of the effect size and the p-value and the test of homogeneity or heterogeneity. Another plot that can be generated from meta-analysis is called the funnel plot. It is a graphical display of the effect size estimate plotted against the standard error or the sample size. The plot will assess the publication bias in the meta-analysis. To do meta-analysis in Jamovi, here I had inserted data from 10 independent studies addressing a specific question. In the first column, the study numbered from study number 1 to study number 10, and the year of publication is given between two brackets. And the next column is the number of participants. So this research study has been designed to have two groups, the treatment or experimental group and the control group. So this column is the number of participants in the treatment group, and then the mean for the treatment group is provided here, and the standard deviation for the treatment group. And for the control group, the number of participants is recorded, and the mean for the control group and the standard deviation is also scored and inserted here in these columns. So here we have an outcome which is continuous, and the means, one of the parameter for that continuous variable is the mean. And there are certain options that we can select to compare and do statistical analysis in those combined studies, either looking at the mean or the standardized mean difference, or using an effect size such as Hedges G. To do the meta-analysis in Jamovi, you need the function for that, and if it is not installed meta-analysis function, then click on the modules here and then click on Jamovi and select the meta-analysis options from here, and then install that. Once it is installed, click on the major and scroll down. Since our data here, it is not correlation, it is not binary outcome, it is size effect, and we will select the mean difference, as we have the number, the mean, the standard deviation, and then we could add more modification subsequently during the analysis. If we click on that, A new window appear here, and for the mean differences, and we need to move each specific variable or data into the right section. So the study or the name of the studies or the list of the studies goes for the study labels, and then for the group number 1 and group number 2, so we have here 2 sections, so we'll move the number of cases to the first box, the mean for the group 1, and then the standard deviation. So these all the data for the first group. For the second group, which is the control, move the number of cases to the group 2 sample size, then the mean to the group 2, and the standard deviation for the control for the group 2. If we stick to the estimate effect size, which is the mean difference, 2 tables are generated, and the model is selected as a random model, and we have 10 studies here, k equal 10, and the important one, which is the effect size, which is the estimate, is 0.73. This is the overall effect size, and it is significant as it has a p-value of less than 0.001, and the confidence, 95% confidence interval, is for the lower limit and for the upper limit, and the estimate lays between these two limits. So there is a statistically significant difference between the treatment group compared to control, as the overall estimate effect size is 0.7. Looking at the heterogeneity between the studies, a test has been done, which is the I-square value is generated, and the value is 20.1%, which means that there is minimum heterogeneity between the studies. We need the p-value to be greater than 0.05, and here it is 0.3. So there is to a certain level minimum heterogeneity between the studies. If we scroll down here into these options that are available, we could also select some of the options. For the model estimator, we could go for the Hedges G and select that, and that will also generate a few modifications. So we could stick to the one which is for the Hedges G value, and for effect size model measures, we'll keep it standardized mean differences, and no changes for the moderator type, confidence interval 95%, display model fits, no need for that. Let's go and tick on the plots. So we need to draw now 2 plots, the forest plot and the funnel plot. And if we scroll down here, we'll see that a forest plot is generated, and on the left hand section is the studies from 1 to 10 with the year of publication, and the dotted line here is the line of no effect, which is 0, and the square symbol here represent the in this case the effect size, which is the Hedges G or the standardized mean difference, whichever option you prefer to go for. And as shown here, each square have different sizes, and that size is larger square, small square. That size of the square, which is the effect size, reflect the sample size. Larger sample size will have larger square, and it will have higher weights percentage influence in the analysis. The horizontal line here for each study represent the 95% confidence interval, and the diamond shape is the overall estimate effect size, which is in this case is given as 0.72. looking at the right section here, each study will have the effect size, which is in this case Hedges G, for every individual study is calculated with the confidence interval. From the forest plot, the sections are here given for the diagrammatic representation, the statistical description, and for homogeneity test is given in a separate table. Here for the forest plot, no major changes will be done, only if the interest in changing the sizes and colors of the effect size point, or the study order, or whether the forest plot is size is small or big, or even I can go and change the effect size point into, let's say, empty squares, and this will reproduce the graph with empty square. Moving to the funnel plot, which is the last section here, as shown in the funnel plot, the plot is have an y-axis and x-axis. In the y-axis is the standardized error, and on the x-axis is the standardized mean differences. And I could go for selecting for the y-axis either the standardized error or the sample size, whichever you find suitable. I will stick with the standardized error on the y-axis, and on the x-axis is the standardized mean difference. Could also select the plot size, whether medium, small, or large. The funnel plot will assess the publication bias. As shown here from the plot, the middle line is the overall effect size, which is 0.72, and the small circle here represent the effect size for each individual study. The effect size study should be evenly distributed above the line for the overall hedge size effect, and the lower part. So the dot here should be evenly distributed in these two sections. Here we have 1, 2, 3, 4, 5, and here we have 1, 2, 3, 4, and there should be no dot outside the funnel plot. So this funnel plot will reflect the whether there is a bias or no bias in the publication. Meta-analysis in Jamovi with an outcome that is binary. A binary outcome or a binary variable is a variable that have two levels in it, such as yes or no, smoking, not smoking, have cancer, doesn't have cancer, dead or alive. So it is a binomial data, only two categories, and the effect size is measured through the odds ratio. Here in the Jamovi data view, I had inserted data from 12 studies as shown here, study ID, and the studies are numbered from 1 to 12. And in the other columns, we have here data for intervention group and data for control or reference group. So this study were designed to have two separate groups. One group is experimental or treatment or intervention, and the other group is the reference or the control group. And the event in each group is recorded as a success or failure, whether the treatment was successful or failure, and the number of participants and the count or the number of participants who have be recovered from the conditions they are suffering using that treatment is recorded here and given a value. So here we have for the treatment group 3 variable, the T for treatment success, number of event of success, number of failures, and the total number of participant. And the same for the control group. The control has not been given the treatment, and the happening or occurrence of the event, which is recovery from the condition, is recorded, and failure to recover is also recorded. So this is event and this is no event, and the total number of participants in the control study is given here. To determine and pull out the overall effect size, in this case, since we have here a binary outcome, the outcome which is either success or failure of the treatment, treated or not treated, so then the outcome is binary or binomial, and the effect size measurement will be here taken as the odds ratio. And to do the meta-analysis to find out whether the data from all these 12 studies agree or disagree on the final effect size, then meta-analysis is performed. And the way to do meta-analysis is to click on analyze, and then if the icon for major is not installed in Jamovi, then click on models or modules, and then select the Jamovi library and look for the meta-analysis icon here and install it. So this is major here, icon is already installed, and we are going to perform meta-analysis using a dichotomous model. Dichotomous mean binary, which only have either 2 levels or categories in it. Click on it, and then a new window will appear here, and we need to move the variable to each section. So here in the number of incident in the experimental or treatment group, which is the total success, and the number of incident in the control group, which is the control success, and the total number or the size sample for the experimental group, which is the T total. Move it to this section, and for the total sample size for control group is the C total. Move it to this section, and for the study label, move the study ID to this section. Simultaneously, few tables are generated with description and a forest plot and a funnel plot. Before we look at the tables and plots, let's take some of the options here. So for the model options, there are certain estimator can be used, so we'll stick to the restricted maximum likelihood. For model measures, there are a few options whether to go for log odds ratio, log risk ratio, risk differences, and so on. So we'll stick with the log odds ratio. No moderators here is selected, and this is 95%. And for the plot, if we scroll down to the forest plot, be no major changes here. And for the funnel plot, as shown here, we'll stick with the option of standard error for on the y-axis. Also, although we can plot the sample variance, sample size, square root sample size, and so on. Back again here to the outcome or the result. The first table shows that the model selected is the random effects model for K, which is 12 studies, and the odds ratio, this is the overall odds ratio for the 12 study, is 2.79, which indicates that there are more recovery in the treatment group compared to the control, so the drug is effective. That is also confirmed by p-value less than 0.05. and standard error is given here, and 95% confidence interval is given here for lower and upper bound, and the estimate is fitting within this range. So these two values, the p-value and the 95% confidence interval, confirm that this observation, the odds ratio 2.8 for experimental related to control, is significant. Moving to the next section here, or table, and this is for heterogeneity test. There are certain tests that can be used, the tau or tau squared, Q. If we stick with the I squared here, it shows that the heterogeneity value is 12.2%, which is a very low heterogeneity, and the p-value is greater than 0.05. We need this p-value to be higher than 0.05. So this confirms that there is minimum heterogeneity among the result extracted from these 12 studies. Description is given here for each one of these, which is the Q test as given here, which Q test is a chi-square. And if we scroll down to the forest plot, which is a very important and a vital diagram that is generated from the meta-analysis. So in this diagram there should be three sections. The first section is given here, a table for overall effect size and a table for heterogeneity test, descriptive data and diagram. So this diagram shows that the study 1 to study 12 on the left hand side, and the model selected is random effects model, and on the right hand side is the odds ratio for each independent study, with the 95% confidence interval for the upper limit, lower limit, upper limit for all the 12 study. And on the section here, this is the overall odds ratio for the 12 studies with the 95% confidence interval. And as shown here from the diagram, the odds ratio fit between the lower and upper bounds of this 95% confidence interval. The section in the middle here represent the studies. The square, small square here, different sizes of the square represent the effect size, which is the odds ratio for each one of them as given here on the scale. For the first study, the odds ratio is 3.8. It's nearly here, 3.8. We notice that there are variation in the size of the square, which reflect the sample size. Studies that have larger number of participants will have larger square as the percentage weighted for the influence of the size is considered. The horizontal line here is the limits for the 95% confidence interval for the upper and the lower. And the diamond shaped symbol here is the overall effect size, or the overall odds ratio, which is 2.8. So this is the essence of plotting forest plot, to make values and to generate values for the odds ratio from all these 12 studies. And it is with confidence can be said that comparing and combining all the results or data extracted from all these 12 studies, there is a significant effect or association between taking the drug relative to not taking the drug. So the drug is effective in curing or recovery from the condition. Now that is the conclusion that can be extracted or generated from this forest plot. Now, moving to the second plot, which is the funnel plot. The funnel plot, as shown here, is a scatter plot in a shape of a funnel. On the x-axis is the odds ratio for each individual study, as represented by black circles here. And on the y-axis is the standard error. And the line in the middle here is the overall odds ratio, which is 2.8. And this plot will show whether there is a publication bias. For no publication bias, the distribution of the effect size for each individual study should be equally distributed above and below the vertical line here, which is the overall effect size. So here we have imbalanced number of studies distributed on the right hand side, or above the OR, overall OR, relative to the below the OR. So there is some degree of bias in this publication, but that ought to be confirmed by further statistical tests such as Eagers test. Meta-analysis with a binary outcome based on an estimate of effect size, which is the risk ratio, abbreviated as RR in Jamovi, and the risk ratio is the ratio of the probability of an event happening or occurring in a treatment or intervention or exposure group compared to a control or unexposed or reference group. In this Jamovi data view, I had placed 12 studies, and the data from these studies in the first column here after the study ID is the intervention group or treatment group. There is an event count and then no event count, and the total number of participants for the group of treatment, and then the control group, an event count, no event count, and the total number of participants. So each study was designed to contain two groups, intervention or exposure and an exposure group, and the happening of the event was measured, counted, and placed in each section corresponding to the study. So here we have an outcome of a data for each study that have two levels in it. So the outcome is binary. There are two levels, either an event happening or no event happening. So this is a binomial dichotomous outcome. And to perform meta-analysis and to find out and calculate the overall effect size, which is the risk ratio, the way to do that in Jamovi is to click on the major icon here. If the package for the meta-analysis is not installed, you can install that by clicking on modules here and then select the meta-analysis package and then install it and then use it. So since our data here is dichotomous, which is binary, select the meta-analysis. Since the data here are binary, or the outcome is binary, then we will perform binary meta-analysis. In this window here, move the variables on the right section. Let's move the study ID into the study label here, and here there are two sections, one for the experimental and one for the control. So move the number of incidents or events in an experimental group, which is the T for treatment events, and the number of incident in the control group, which is the C control events, and the total number or total sample size for experimental group is the T total, and for the control C total. Here we moved all the relevant data to perform binary meta-analysis, and as shown here, two tables showing the outcome. The models that is selected is the random effect model. This model can be selected from the option here, clicking on the options for model and then select the model estimator, and either go for random or fixed. We'll stick with the random effect model using the restricted maximum likelihood. The model measures is the log risk ratio. We'll move that into risk ratio as we are going to calculate the risk ratio here. We're not doing the odds ratio. So for the risk ratio, and then no changes here. So two tables are generated as shown here. The model is a random effect model, and the pooled or overall estimate of the effect size, which is the risk ratio, is generated, which is 0.85, and the p-value less than 0.05, which is significant, and a 95% confidence interval, which ranges from 0.6 to 1.06. So both the p-value and the 95% confidence interval had confirmed that this pooled risk ratio is significant. The next table is the test for heterogeneity among the results, and the Q statistic is performed, which is based on chi-squared, and the p-value is generated, and an I-squared is also produced, and the I-square is 21.2%, and the p-value need to be higher than 0.05. So this indicates low level of heterogeneity among the results. The output also will generate a forest plot and a funnel plot. So for the forest plot, click on the section here, and this one will be ticked, and there will be no major changes here. Looking at the forest plot, as I have described, there are three sections. The first section is descriptive, the second section is diagrammatic representation, and the third one is the overall, which is given in a separate tables. So here the list of the studies from 1 to 12 is given here, and this figure here is the risk ratio for each individual study, and the 95% confidence interval, and the overall risk ratio is given in this area here, which is 0.85, and a 95% confidence interval is calculated, and a scale is given here. As shown in this diagrammatic representation, the small squares represent the risk ratio for each individual study, and the diamond shape symbol is the overall risk ratio, and the horizontal line is the limits for the 95% confidence interval, and the size of the squares varies depend on the size of the sample, and the dashed line here is the line of no effect. We could also do some editing on the forest plot if we wish, by changing the shape of them into circles, or we retain them as a square. So there are certain options that can be selected. For the final plot, which is the funnel plot, the y-axis can be plotted in it either a standard error or sampling variance or sample size or others. The plot, which is the funnel plot here, as shown in the x-axis, is the log risk ratio, and on the y-axis is the standard error, and the plot is a scatter plot in a shape of a funnel, and the circles here represent the risk ratio for each individual studies, and the line in the middle here is the pooled effect size, which is the pooled risk ratio, which is about 0.85. And from that funnel plot, it can be said that there is publication bias, as the distribution of the risk ratios has to be symmetrical along above and below the line in the middle, as here we have 1 2 3 4 5 6 7 8 risk ratio studies with the risk ratio above the line, and 3 below the line, and 1 outside the funnel. So further statistical tests need to be done, which is the Eagles regression test. Proportional meta-analysis in Jamovi and generating a forest plot and a funnel plot. Proportional meta-analysis is a single group type of meta-analysis which is different from pairwise group meta-analysis. The proportional meta-analysis is a statistical method that combines results of different studies based on the effect size estimate, which is expressed in percentage or proportions or rate or frequency. The proportional meta-analysis will calculate the pooled overall estimates of proportion or rate from several independent studies addressing the same scientific, medical or clinical research question. So for the meta-analysis to be performed, the proportional meta-analysis, the data have to be recorded in a format of proportion or percentage or rate. So any binary data reported in percentage or rate or proportion can be included in the meta-analysis. This type of meta-analysis is commonly used for the analysis of prevalence and the incidence of conditions or disease in population or a group. The proportional meta-analysis will also examine and assess the degree of variability and inconsistency among the results from several independent studies by performing a heterogeneity test based on Q statistics, which is based on chi-square, to look for differences or variation in the results, which is a measurement for heterogeneity, and a value of I squared is generated, which is represented in percentage, and the 0% indicates that there is no heterogeneity, and the 100% indicates maximum heterogeneity. Here in this Jamovi data view, I had inserted 12 studies from the literature and the data extracted from the studies. Each study was looking at the incidence or the proportion of a specific condition in a certain group, and the number of the events occurring were reported, and the total number in the study of participants were also included. Here we have a type of data that is in a shape of or format of proportion or a percentage or a rate, and it is only single group, no comparison between control and intervention or exposure. So here we will use the proportional meta-analysis to combine all the data from those 12 studies and then to calculate an overall or pooled proportions or rate. And the way to do that in Jamovi is to click on the major package. If you have not installed that, install that from the modules here by clicking on the module and then Jamovi library and look for the major and install that. Click on major and then select the meta-analysis for proportions. A new window will appear here for proportions. Move the study ID into the study label and the proportion, which is the events, into the frequencies of the events, and the sample size, which is the total. Simultaneously, few tables and graphs are generated or plots as shown here. By clicking on the model options, we have here options to select from, whether to stick with the restricted maximum likelihood or Hedges or DerSimonia, Laird, or any of these estimator, and we'll stick with the first one. And for the effect size, the model will measure, we could select the raw proportion, log transform proportion, or log transform proportion will keep the raw proportions. Confidence interval 95%, and for the plot will keep the plot now and look at the tables generated here as shown from the two tables. The first table shows that the random effect models was chosen and the total number of studies were 12, and the estimate of the effect size, which is a proportion, is calculated and found to be equal to 0.34, and a p-value is less than 0.05, and the 95% confidence interval with an upper limit and lower limit that contains the pooled estimate proportion. So this data shows that this proportion is statistically significant as confirmed by the p-value and the 95% confidence interval. The next table shows the homogeneity or heterogeneity statistics, heterogeneity test, in which an I-squared value is generated and a p-value is generated based on the Q statistics, which is a chi-square. The I-squared value is nearly 70%, which is very high, so this indicates that there is high heterogeneity among the results. However, the p-value here has to be higher than 0.05. Moving to the next section, which is the forest plot. Drawing the forest plot, there are certain options to select from. I will not perform any changes here. We'll stick with the default one as shown here from the plot. Should have three sections. The third section, which is the overall and the heterogeneity, is given in separate tables in Jamovi. And in the first set here is the studies ID from 1 to 12, and random effect model is selected. And on the right hand side is the proportion calculated for each individual studies, or the percentage, and the 95% confidence interval for each study. And also the overall proportion is also calculated with its 95% confidence interval for the upper and lower values. The middle section here, which is a diagrammatic representation of the effect size, which is the proportion. So each square here represents the proportion or the percentage or the rate for each individual study. There is a variation in the size of this square, and that reflects the variation in the sample size. Larger sample size have larger squares, as shown here for this one, and for this one is a small square, which indicates smaller sample size. The horizontal line here represent the 95% confidence interval, and the scale here range from 0.1 to 0.6. The diamond shaped here represent the overall calculated proportion or pooled proportion. And if we scroll down here for the next section, which is for publication bias can be assessed using statistical method, or it can be assessed using a plot by drawing a funnel plot. And as shown here in the funnel plot, it is a shape of a funnel with an x-axis and a y-axis. On the x-axis is the proportion for each individual study is plotted, and on the y-axis is the standard error, and in the line in the middle represent the pooled proportion. And as shown here, for no bias in publication, the distribution of the effect size, which is the proportion, should be equally distributed on the right side and left side of the middle line here. So the number of proportion, which is 1 2 3 4 5 on the right hand side, and then 1 2 3 4 5 on the left hand side, and there is one outside here and one outside here. This funnel plot will validate whether there is a publication bias. And another statistical test, which is Eager's regression test, required to be performed to validate whether there is a bias or not. Meta-analysis with continuous outcome based on an effect size, which is correlation, or R for correlation coefficient, in Jamovi. Here in Jamovi data view, I had placed 12 independent studies that addressing the same scientific or medical or clinical question, and data are collected from each study, and the total number of participants recruited to the study were also recorded, and the correlation coefficient, which is an effect size, was calculated for each study. To perform meta-analysis based on an effect size, estimate of effect size, which is a correlation coefficient, which is R here in this case, to calculate the pooled overall correlation coefficient, and to draw a conclusion based on those 12 studies. To do meta-analysis in Jamovi, first of all, download the package for meta-analysis if it is not installed by clicking on modules and then scrolling through the library, Jamovi library, and then download the major package. Once installed, click on the major package and select the meta-analysis for correlation coefficient with an R and an N for the numbers or total numbers. A new window will appear, and there are sections here for correlation coefficient. Move the study ID into the study label, and then move the R into the correlation section and the total for the sample sizes. Simultaneously, a few tables are generated with description and a forest plot and a funnel plot. So if we tick on the model option here and scroll up to the top, the model estimator here, there are several options to select from. I will stick with the restricted maximum likelihood, and for the model measures there are other options. We'll stick with the Fisher's R to Z transformed correlation coefficient, and no moderators here, and the test will retain as Z test and 95% confidence interval. For the forest plot, there will be no major selection, and for the funnel plot, simultaneously no selection. And for publication bias, there are few selection, but we will keep the default format as it is assigned, and for additional options, no selections. In the output here for the analysis, as shown from the first table, for the correlation coefficient for meta-analysis, a random effects models was chosen, and there are 12 studies. And the estimated effect size, which is the correlation coefficient, is 0.5 with a p-value that is significant less than 0.05 and a 95% confidence interval that contains or includes the value for the pooled correlation coefficient. And a second table for testing the heterogeneity, and as shown here, the I-squared is produced, which is 4%, which indicates very low level of heterogeneity among the results, and the p-value greater than 0.05, and the q statistics based on the chi-square. Scrolling down to the forest plot, as shown here from the forest plot, the ID for the 12 studies is recorded on the left hand side, and the random effect model is recorded here. The plot should consist of three sections: descriptive sections, a diagrammatic representation of the descriptive statistics, which is given here with the correlation coefficient assigned for each study and a 95% confidence interval, and here toward the lower end, overall correlation coefficient is calculated to be 0.5 with 95% confidence interval. The small or variable squares here represent the correlation coefficient for each individual study, and there is a variation in the size of the square, and that is reflection or representation of the sample size. Larger sample size will have larger square, as it will have higher influence or impact on the overall meta-analysis. The horizontal line represent the 95% confidence interval, upper and lower limits, and the dashed line here is the line of no effect. And the diamond shaped structure here represent the overall or the pooled correlation coefficient, which is 0.5, and a ruler for the scale is presented here at the bottom. Moving to the other sections here, further tests are performed as shown here for publication bias assessment, and Eager's regression was performed as given in this table, and a p-value is assigned for this Eager's regression. Scrolling down to the funnel plot as given here, the funnel plot is a scatter plot that has on the x-axis the correlation coefficient for each specific studies as represented by the black small circles, and on the y-axis is the standard error, and the scatter is in the shape of the funnel, and the line in the middle is the calculated overall correlation coefficient, which is 0.5. And for lack of publication bias, the distribution of the effect size, which is the correlation coefficient for each individual study, should be symmetrical or distributed evenly on both sides of the line, above the line and below the line. And here we have 1, 2, 3, 4, 5 on the right hand side, and 1 2 3 4 5 6 7 on the left hand side. So this funnel will determine whether there is a publication bias supported by the Eager's regression test outcome. If you have a data and you want to perform data analysis and you are looking for one-to-one private tutoring sessions or a training course on how to analyze the data using any of these packages, SPSS, Stata, Jamovi, Statistics, GraphPad Prism, or any of the others, then do contact me.